It's a real pleasure for me to be able to introduce our next speaker, Dr. Jerry Peer, who is currently professor of medicine particularly in the microbiology and molecular genetics department at Harvard Medical School. Jerry grew up in Los Angeles, so welcome back to your state, and also welcome back to Berkeley, where you got your PhD after attending the University of Pacific in Stockton, California. He got his PhD in microbiology, spent a lot of time in the building that's no longer here, called Warren Hall, and did his degree work with Stuart Madden. And was awarded that degree in 1976, and I understand we're going to see a picture of him from that period of time during his talk, which will explain why it took him 12 years to get his PhD in the 70s. So the major focus today of his professional work has been the role of bacterial capsules in pathogenesis as antigenic targets for vaccines. He works on a broad spectrum of both gram-negative and gram-positive organisms of clinical significance, including those that produce a particular polysaccharide, Poly-N-Acetyl Glucosamine, which we'll hear a lot about today as a potential vaccine target. Jerry?
Thank you very much, Mike, for the kind introduction. As some of you who are here for the panel know, we had some discussion about conflict of interest and disclosure, so I do have to make some disclosures as part of my conflict of interest requirements regarding the technology I'm going to discuss today. So I do have sources of income from two companies that are currently developing both the vaccine and a monoclonal antibody to this antigenic target, Poly-N-Acetyl Glucosamine. And now that I've disclosed it, you can be sure I'm completely unbiased in everything I have to say here. So as was mentioned, I did get my PhD here. It actually only took me 4.5 years. And I'm not much of a picture taker, but there was one picture taken fortuitously. Is my microphone not on? Okay. And it was a funny development in that I was called about an hour before the graduation ceremony, and I really hadn't even started writing my thesis, but was told I had to march in the graduation line for that particular graduation. I was going to be gone within a year, and I was the only PhD candidate, so they called me up. Of course, I had no cap and gown and hadn't even intended to attend the ceremony, but I called my girlfriend at the time. She came running over with a camera, and we were able to get this picture of me, that's me right there, getting my PhD in the eucalyptus grove, which I see is still here. And here's Trudy Buehring, who had just started as a young faculty member and is still here. Behind the microphone is Sandy Elberg. He was the dean of the graduate division from about 1962 to 1968. He passed away at the age of 97 just a couple years ago. And over here you can see the great statistics professor, Dr. Chang, who just died last April at 99. So I'm really hoping this age and survivability rubbed off on me a little bit. And, you know, I walked past the eucalyptus grove this morning and I noted that, you know, it seems to have the same purpose as it did back then by the sights and particularly the smells that one encountered there. I did get out of that a degree, and this is hanging in my office. And notably, the signature is by one Edmund G. Brown, Jr., Governor of California. And I kind of wonder, do you have elections in this state? You know, things don't change much, do they? Anyway, I could tell a lot of other stories, but I should talk about the topic at hand. So I want to talk about bacterial, and this is really, I haven't changed all my slides to microbial, but this could say microbial strategies to avoid surface antigen-specific immunity. So this is one of the major challenges that everybody in the room probably knows about for vaccine development, is on occasion we get an organism with a very limited serologic diversity. We can make a vaccine with one antigen, or maybe 2 or 3 antigens, or 2 or 3 attenuated strains. But a lot of organisms have been much more difficult to develop vaccines against for a variety of reasons. Now, of course, one of the major ones is they produce lots of different target antigens. These are often surface capsules or LPS antigens on gram-negatives. So strep pneumo is the champ, but a lot of the gram-negatives, E. coli and Klebsiella pneumonia. Some, such as the meningococcus and group B strep, they don't make tons, but they make a fair number of target antigens that makes vaccine development challenging. Some microbes make major target antigens, surface capsular polysaccharides, that aren't immunogenic in the population at greatest risk. So H. food type B, E. coli K1, and the meningococcal serotype B, the latter two are self-antigen, so they don't induce immunity. The H. food type B, we solve that problem by making a conjugate vaccine that's immunogenic in kids under the age of 2. And there's a third category, somewhat less studied, but typifies the antigens I've been interested in. And these are molecules that are immunogenic. They induce natural antibodies. They're surface polysaccharides, but the antibodies they induce are not opsonic and not protective. And therefore, the organisms are able to make these antigens, and the host makes an immune response, and the organism doesn't care. So, the molecule I want to talk about is this guy today, PNAG, or Poly-N-Acetyl Glucosamine. It's a pretty simple molecule. It's only got one sugar, N-acetyl glucosamine. The two monomers have this beta-1,6 linkage, for those of you who remember back to carbohydrate chemistry. That's pretty simple. There are some acetates and succinates that are linked by hydroxyl groups. Well, they're not very well studied. We don't know very much about their role or anything. And then most of the amino groups that you can see here are acetylated. There are a few free amino groups on the molecule. This is pretty important for gram-positive organisms. They need to strip off about 5% of the acetates in order to get the polysaccharide anchored to the cell surface. If they don't do that, they make the polysaccharide, but it doesn't stay on the surface. In gram-negatives, it's an essential function. And the genetics and chemistry behind synthesis of PNAG is actually becoming very difficult and challenging to define in terms of the fact that it's very different among all these different organisms that make it. So the genes were first discovered in Staphylococci in Fritz Getz's group in Tübingen, Germany. They identified 4 genes that they just called ICA-A, D, B, and C. D was discovered later. And 2 of these genes, A and C, are involved in biosynthesis. They put the polysaccharide polymer together. The D gene is involved in polymerization and secretion. It helps form a channel. And the B gene strips off some of those acetates from some of the amino groups on the glucosamines. The factors that were found in staph to affect transcription and polysaccharide production, it was produced in higher levels if there was glucose, ethanol, increased salt. Some strains do better under low oxygen. This also means that the ideal growth medium is a margarita. And there is a divergently transcribed negative regulator, and when this is activated, transcription of these genes is repressed. So that looked all kind of interesting and well and good. And then about 8 years later or so, it was discovered that the genes were also in E. coli, but it wasn't really the same genes. By kind of the typical bioinformatic analysis available at the time, the E. coli genes didn't look at all like the staph genes. It was only after Tony Romeo and colleagues, who was at Emory at the time, were able to define via this sort of saturated transposon screen these 4 genes in E. coli, and what they were looking for actually was loss of the production of biofilm in vitro, because PNAG production in staph had been primarily associated with biofilm formation. So these 4 genes, PGA A, B, C, and D, form an operon in E. coli. These are relatively well conserved in other gram-negatives. Not all gram-negatives, but a lot of gram-negatives have this locus. And once this was sort of discovered, you could sort of see the similarity with the gram-positives, but it's not very high in terms of protein structure or amino acid sequence of the proteins in particular. Now, 2 models have been proposed for synthesis of PNAG in gram-negatives and gram-positives. This came out of Tony Romeo's group as well. So in the cytoplasmic membrane, PGA-C and PGA-D form a heterodimer. This is cyclic GMP dependent, so they have cyclic GMP binding sites. And they appear to initiate polymerization from the precursor, UDP-N-acetyl glucosamine. This protein, the ICAB protein in the periplasm, both binds to the emergent polysaccharide and deacetylates it in part, and then PGA here helps get it out through the outer membrane. How this is linked to the cell surface is still not known. In gram-positives, this is work that was recently published by Lynn Howe's group out of Canada with her postdoc Dustin Little. And what they've shown is that you have similar structures. So you have ICAA here, which is actually more equivalent to PGA-C, and pairing with the ICAD protein to form a polymerization and secretion channel through the membrane of a gram-positive. ICAC, it's a little mysterious. It does have some enzymatic function and seems to promote polymerization. And ICAB is outside the membrane here and below the peptidoglycan. They discovered this very small tail down here, you can barely see it, to anchor it to the membrane. And this does perform the N-deacetylation, and it may also be involved in adding those succinates and acetates to the hydroxyl groups. You can, as I said, eliminate ICAB from the gram-positive, but you need all 4 genes in the gram-negative. Okay. So we started looking for organisms that made PNAG, and it was first, as I said, discovered in Staph epi, then we found it in Staph aureus, and then Tony found it in E. coli. And this is a list of organisms where the genes have been found and knocked out, the polysaccharide's been isolated and characterized 100% of the time so far. It's been authentic PNAG. So it's hard to isolate PNAG, particularly if you don't overexpress the biosynthetic genes. But these guys make the polysaccharide and they react immunologically with our reagents. This is a second list, and you can see it's growing. Genes have been identified. When we or others have knocked them out, they've lost the reactivity with our immunologic reagents, our antibodies to PNAG. Many, but not all of them, have been complemented and production has been restored. And you can see the list is now growing with a fair number of interesting organisms on there. We just finished characterizing the B subtilis genes and polysaccharide and proteins that are involved in PNAG synthesis, and they don't look anything like any of the other ones. Oh, and also in strep pneumo, we just identified the genes. They don't look anything like even the staph ones. So it's a big mystery. And then this much longer list here of microbes that make PNAG, but we've only shown this by immunologic confirmation. And I'll show you how we do that in a bit. Now, some of these we've also put through animal models, or others have. What became very interesting here was the fact that we started to look beyond just bacteria. And we found fungal pathogens, Candida, Cryptococcus, and several others that make PNAG. We found it's on malaria, both mouse and human malaria strains. We found it's on Trichomonas vaginalis, another protozoan pathogen. And very recently, a collaborator in Brazil who we sent antibodies to have found that trypanosomes and toxo are positive for PNAG as well. So it looks like this may be a very broadly expressed antigen. The question is it going to be a vaccine target? Okay. So how do we detect PNAG? Well, this is PNAG with its beta-1,6 linkage in comparison to chitin, which has a beta-1,4 linkage and is the second most abundant polysaccharide in biology. PNAG can be digested by this enzyme, dispersin B. It's been crystallized. It's highly specific. It doesn't touch anything else, including chitin. It can also be totally degraded by periodate because it's got these periodate-sensitive bonds, whereas chitin basically can be digested with chitinase, but it's periodate-resistant and dispersin-resistant. So the way we define an organism as being PNAG positive is by treating the cells with this with either chitinase, to which PNAG is resistant, dispersin or periodate, and reacted with this human IgG1 monoclonal antibody that we have, or a control antibody to Pseudomonas alginate. And if we get reactivity with chitinase treatment, but it's lost with dispersin and periodate, that's our criteria for saying something makes PNAG or PNAG-like organism. So this is what it looks like with staph. This is the control antibody. Up here, no reactivity. The red shows DNA staining. This is the antibody to PNAG directly conjugated to Alexifor. You see they light up quite nicely. This is the overlay. And then after dispersin or periodate treatment, the reactivity with the monoclonal goes away. Now, I'll be the first to tell you it's very dangerous to do structure by immunologic cross-reactivity. But when we add in the dispersin and the periodate, we're fairly confident that this is something that has PNAG in it, on it, or part of some other structure. We've yet to find this reactivity not associated with PNAG. That doesn't mean we won't, but it's more robust than just antibody binding. So just a couple of examples here. I just chose to show you some pictures of yeast, but we have them for Plasmodium. We have them for most of those bugs. Here you can see Candida albicans in either the yeast or the hyphal form, or A. flavus, and you can see this is the control mab, no reactivity, DNA stain, the overlay, and the antibody to PNAG over here, you can see the yeast cells light up and the hyphae light up. And if you come down here, if you take the antibody, F598 is to PNAG, you can see that the yeast or the hyphal forms of Candida or A. flavus light up, and then when we treat them with dispersin or periodate, the reactivity goes away. So we've seen this for lots of different organisms, and so we're pretty sure this does represent PNAG. It also turns out that it is on the surface as a polysaccharide, and it functions as a capsule. Now, the definition of what's a capsule for a bacterium is really pretty loose, and there really is no good definition. It's just something that reacts immunologically with an antibody that by electron microscopy looks like it's on the bacterial surface. Nonetheless, two organisms that have traditionally been felt to be unencapsulated, the gonococcus and non-typable H. flu, in fact, do have PNAG on their surface, as you can see here. You know, the staining's not always all that robust. Sometimes it's very heavy, sometimes it's not. But it does look like what we traditionally call a capsule for these traditionally unencapsulated organisms. More importantly, it's also present on the surface of N. meningitis serogroup B, and this is that one strain of meninge that you all know we don't have a good vaccine for based on the capsule because it's not immunogenic, so this is a potential target. And then with meninge serogroup A, what I want to show you here, as well as with strep pneumo down here, here's meninge serogroup A reacting with antibodies to PNAG, with antibodies to serogroup A. And if you come over here and see this magnification where they were dual stained by 10 and 15 micron particles, you can see that the PNAG is intercalated on the bacterial surface with the traditional capsule. And the same thing's true in streptococci as shown down here. So it's in the same physical space as capsules, and that's good news because capsules so far make up some of our best antibacterial vaccines. Now, another critical factor, of course, that you want to know is whether or not the antigen is expressed in vivo during infection. And so I threw this in here because we're talking a lot about TB. And in a paper that we published in PNAS about 1.5 years ago, we in fact found PNAG, as you can see here, associated with TB antigens in the lung of an infected human. So you can see the various categories. The DNA stain here is blue. This is the control monoclonal, it doesn't light up. But here's different fields where you can see either something that looks like a tuberculous, a pile of TB antigens, this is antibody to TB, or another pile of antigens where there's some PNAG around it, but not necessarily on it, and it doesn't really look much like a TB bacillus, but nonetheless, we can gather evidence that the PNAG antigen is expressed in the lung of an infected human. We've shown similarly for strep pneumo and non-typable H flu and middle ear fluid of kids, in the Staph aureus in the lungs of CF patients, and a number of animal models that the antigen is expressed in vivo. Okay. So given the broad expression of PNAG, and if you haven't figured it out already, a lot of those organisms are normal flora, why aren't we all walking around with lots of antibody protecting us from all these pathogens? Okay, that would make a lot of sense, and evolution is usually pretty efficient. Well, it turns out we all do have a lot of antibody. Everybody we've looked at is an individual, or I'm showing you here data for pooled human sera, or IVIGG, which has a 5 times concentration, so the curve's only shifted higher because it starts off with 5 times more antibody. We get very nice binding to PNAG antigen and ELISA. Most people, like this pooled serum, have an endpoint titer of around 1 to 1,000, 1 to 2,000. And this antibody in human serum, normal human serum, is all IgG2. Now that's typical of the subclass of IgG that we make to carbohydrate antigens. However, the natural antibody elicited to PNAG by natural exposure are generally not opsonic and not protective. So we've screened about 65 individuals to date, and only in 4 of the individuals were we able to detect PNAG antibody that was functional in an opsonic killing assay. So we would test the antibody's ability to kill staphylococci, show it was PNAG specific by absorption studies, and very few healthy adults have functional antibodies. So most of us have antibody, but it doesn't work to mediate killing in the lab, and it doesn't work to protect against infection. This is a mouse skin infection model with Staph aureus, and we compared the ability of normal human sera with a good titer to PNAG of IgG2, normal rabbit serum or just PBS, and you can see there's no real difference in the bacterial level in the skin of mice 72 hours after infection. So a natural antibody to PNAG just doesn't have much in the way of in vitro or in vivo properties associated with protective immunity. So how do we go about turning this into a vaccine? Well, I'd like to tell you I am this clever genius that figured this out, but in fact, a sort of traditional way of looking at polysaccharide antigens and their immunogenicity is to ask about the role of the acetates. Okay, this goes back to the development of the meningococcal vaccine in the 1960s. So we took a look at PNAG and we chemically treated it to remove most of the acetates. So only about 10 to 15% of the acetates were now acetylated. We refer to this as dPNAG for deacetylated PNAG. And using this as an antigen, we were able to show we could now get in vitro opsonic antibodies and in vivo protective antibodies. So we started out by immunizing a couple of rabbits. These 2 rabbits got the native highly acetylated PNAG, conjugated to a diphtheria toxin mutant as a carrier. You can see they made a lot of antibody to native PNAG, both of them. And they did make antibody to the deacetylated PNAG, but this is a log scale. So in comparison, this was only 1 to 2% or so of the total antibody. In contrast, when we immunized the rabbits with the deacetylated form, coupled to the same carrier, they made good antibodies to the immunogen, but they also made good antibodies to the native form of the polysaccharide as shown here, which of course is critical because that's what's on the surface of organisms. So if you're not going to bind to what's on the organism, who cares, right? And you can see that now the titers were pretty comparable. More importantly, when we compared these antibodies in vitro in opsonic killing assays, again, this is against Staph aureus, but we've done this many times with many different targets, you can see that the antisera raised to the deacetylated glycoform had much better killing activity and a much higher titer than the antibodies raised to the native glycoform. And you'll recall the actual overall titer to the antigen was higher, but they were far less functional. We then went on to try and develop some human monoclonal antibodies, and I'm not going to give you the whole story there, but we did do that. These antibodies were derived from B cells of an individual recovering from a staph infection, so they're fully human, they've never seen a mouse, they have no mouse components. And we screened for binding to both the deacetylated and the native glycoform of PNAG. And we ended up choosing 3 antibodies to work with. This is the mAb F598 that I showed you before we used for immunologic investigations. It binds to both the native and the deacetylated glycoform. Then this one here in the middle, F628, it binds mostly to PNAG. It has a little bit of binding to dPNAG. And this one, F630, which is this one here, only binds PNAG and not to the deacetylated form. And as we found with the polyclonal sera, the human monoclonals that bound to both native and deacetylated had the best in vitro opsonic killing. This guy was kind of intermediate, and this guy was really pretty poor. So the development of these monoclonals and the F598 has been through phase 1 safety and immunogenicity safety and toxicity trials in humans. So far, so good. Nobody died. It has been in limited phase 2 trials, and we're currently making another batch to start some more phase 2 trials later this year. Okay. Now, the basis for this, as it turns out, isn't terribly insightful or interesting. It has to do with complement deposition. So our best antibody, F598, gives us very good binding gives very good complement deposition, C3b opsonins, onto microbial surfaces. The intermediate antibody is also intermediate in terms of its ability to deposit complement, and the antibody with the poorest opsonic activity and does not bind to dPNAG very well, gives us the lowest amount of complement deposition. This is C3. We get the exact same answer if we look at the binding of C1q to target antigens. So activation via the traditional pathway of complement seems to be a major important mechanism for this antibody's activity. Okay. So we've been trying to figure out what it is about this antigen that makes the non-functional antibody so immunodominant. So working with synthetic chemists in Russia who've made a variety of oligosaccharide constructs, we've looked at the structure of both the deacetylated, this happens to have 7 sugars in it, and there's no acetates on the amino group, or the fully acetylated, the N-acetylated glycoforms. And what they found is, what's commonly found in biology, is the polysaccharide forms a helix. There are about 4 turns, 4 half turns, and about 7 monosaccharides make up a complete cycle here. And if you look at this from both the top and the side views, what's very striking, particularly with the top view, is that the acetates, shown in the boxes here, are all on the outside of the molecule. So when this molecule comes into a lymph node and a B cell is going to be presented with this molecule, what's the major epitope that the B cells are going to see? And the major epitope's going to involve these acetates. So we think that's the reason why those acetate-specific antibodies raised to the natural polysaccharide are so common and the dominant form of antibody that we see in both natural human and animals. A lot of animals have natural antibody to PNAG as well. The deacetylated glycoform you can see is much more varied. Okay, so what we think is going on is that with the acetylated glycoform and the acetate sticking out, when antibodies bind to these epitopes, they seem to line up in a way that the FC portions just aren't that close together. We have some structural evidence for that via NMR from my Russian colleagues, but we have definite evidence for that in terms of C1 binding, where these are very poor at binding C1. And what we think is going on with the antibodies to the non-acetylated epitopes that we can engender with our altered glycoform is they bind at a more angular level, and now the FC regions of the antibodies are much closer together. That's a much more efficient way for an IgG1 molecule to bind to 2 C1q arms of C1, and that gets the classical pathway of complement going. So that's kind of our working hypothesis, and my Russian colleagues say they're working hard on this, but it's not easy to do with the kind of chemical techniques available. Okay. So, in addition to complement fixation, we've also looked at the efficacy of the monoclonal antibody in other settings where we've been able to show an effect, and this is all, of course, in mice. And we've been wondering particularly about the role of PMNs in the mediation of the activity. We can show in vitro that there are gram-negative organisms that we can kill with just the antibody and complement. We always need PMNs for gram-positives, but even for some gram-negatives, we need PMNs. So in 2 papers that have been published relatively recently, we showed in a mouse model of infectious colitis that spontaneously occurs in rag knockout mice. I made a mistake here, they lack the T-bet transcription factor, and they're called truck mice. The monoclonal antibody was actually quite efficacious in reducing pathology, even though these animals have no functional T cells, no functional B cells, and poorly functional ILC cells in the intestine. In contrast, we found in a model of Staph aureus corneal keratitis, and this is work we've been doing in corneal infection since Susie Fleissig, who's now a professor here and my former postdoc, got us interested in corneal infections. There's a lot of interesting things about studying infections in the eye. It's generally regarded as an immune privileged site. I'm not going to get into that too much. But here we found that protection is mediated by microbiota matured CD4 positive IL-17 producing T cells. So in the truck mice, these T-bet rag double knockout mice, we were able to show that if you just treat these mice, which get a spontaneous infectious colitis by 8 weeks of age, the major players that have been identified by Wendy Garrett, who worked on this model when she was in Lori Glimcher's lab, are a Klebsiella, a Proteus, and some anaerobes. And our antibody very nicely decreased the cumulative level of pathology. And the pathology scores are derived by measuring in the tissues after 8 weeks of age these 4 parameters: monocytes, hyperplasia, injury, and PMN. They're done by a rodent pathologist who, of course, doesn't know the source of the tissue. And I think most importantly, what our antibody treatment was able to do was to reduce the injury level to a much greater degree than the open squares, which are the control, and as well as the hyperplasia, which is much lower here compared to that occurred in the controls. We didn't see that much of a difference in some of the cellular infiltrates, but I think since injury and hyperplasia are major measures of colitis, this was very encouraging. So this is in mice with no functional acquired immune system and a compromised innate immune system. In contrast, if we look at the antibody in a corneal keratitis setting, we see with these 3 different strains of Staph aureus, including this strain, which is a classic USA 300 community-acquired methicillin-resistant Staph aureus strain, giving the antibody to the mice either systemically or by applying it topically. It works both ways. We can reduce the level of bacteria in the cornea at 48 hours, as you can see here, compared to the controls. And we also look at the pathology that occurs in the cornea, grading them from 0, uninfected, to 1's a little haziness. But by the time the cornea gets a score of 2, it's become opaque. And this is, of course, the major issue in keratitis and eye infections is the immunopathology that occurs in the cornea, causing it to opacify, which then makes it much less able to transmit light, and that is an underlying cause of blindness. And infectious diseases as causes of blindness are a really major problem, as I'm sure many of you know. However, if we did the same experiment in germ-free mice, so that they really don't have a very mature immune system, now the antibody, as you can see here by pathology score or bacterial levels with 2 different staph strains, really does nothing. So it doesn't have any efficacy when we don't have microbiota-driven immune cellular maturation. If we take germ-free mice and we house them with normal flora mice, they reacquire the ability for the antibody to be efficacious, as you can see here, in both bacterial levels and pathology score. And this is dependent on getting PMNs into the cornea. So what we see is that the germ-free mice corneas look just fine, but in wild-type mice that get the control antibody, they have a lot of infiltration of PMNs, which is what you would expect. The immune mice have a very low level because they're clearing the bacteria. But over here in the germ-free mice, what you can see is that we can get antibody to PNAG on the bacteria. They deposit complement, but there's no infiltration of PMNs either into the monoclonal antibody treated, the anti-PNAG, or the control mice. So these guys should look like these guys, and they don't. So this is dependent on IL-17, as shown here, and it's also dependent on CD4 positive T cells, as shown here. So in IL-17 receptor knockout mice, the antibody doesn't work, and in mice that are depleted of CD4 T cells with an antibody, the antibody does not work in keratitis. So two very different situations. So we have a long list of organisms, as you can see here, done by a number of investigators for which we've shown efficacy of our vaccine or the monoclonal antibody. There's a lot of bugs there. Many of these, as I said, were done by us, but a fair number of these were done by collaborators, so it's not just our data. And we are making a vaccine, and I'm running out of time here. The vaccine that we're making is a conjugate of actually a synthetic oligosaccharide, so it's only got 5 sugars. It's conjugated to tetanus toxoid, and we then get this sort of tetanus toxoid linker pentameric oligosaccharide conjugate. It's immunogenic. It induces complement-fixing antibody, and in a trial that we did, I'm going to go through, you don't need to see this, I'll just explain the data, because that we did in pigs at Cornell Vet School by a collaborator there. They immunized pigs with either the vaccine conjugated to tetanus toxoid or to CRIM-197, that's a cross-reactive mutant carrier used in vaccines like Prevnar. And you can see, although it was only 50% protection engendered by this conjugate, the challenge itself was very rapidly lethal, with most of the non-immune mice dying at 17 to 24 hours. The CRIM antigen wasn't protective either. That was a little curious, and what we basically were able to show was that the antibody to TT engendered much better binding to the deacetylated glycoform, so the pigs that lived or survived longer had better antibody binding to the dPNAG, and for the most part, except for these two guys here, they had better complement deposition. We're not quite sure why these guys weren't protected. We think it might be due to the fact that they may not have gotten C3 on there, and we should do those experiments shortly. So in summary, PNAGs are broadly conserved immunologically as well as structurally antigen on the surface of many microbes. Natural immunity to PNAG is generally poorly effective at mediating immunity due to low complement activation. By coming up with altered and synthetic glycoforms of dPNAG, we're able to induce antibodies at least in mice and now in pigs. And we should know in a couple months about more pig studies in horses, which get a infection with an organism called Rhodococcus equi. It's a TB-like organism, whether we'll really start to expand our efficacy in animals, which are challenged with pathogens that they get and cause harm. But ultimately, how many potential targets of this vaccine will actually be realized? Well, as I tell people, you might say, I heard this talk before, all these bugs we had vaccines for. You might say, whatever happened to that guy? I thought he had a good project. How come he dropped out of sight? And I don't know where we'll be on the spectrum there, but I hope we'll at least be partly on the spectrum. This is a good spectrum to be on, by the way. Where at least some of these organisms we'll be able to target with our vaccine. So there's a long list of people to thank. I won't go through them all, but they all have been very helpful in getting this project to this point, and I thank you for your attention.
Time for questions?
Maybe I'll start. I have 2 questions. So does the amount of PNAG that you see vary depending on what type of sugar substrate you feed the organism?
Well, as I mentioned, in Staphylococci, if you feed it glucose, you get much greater production. Interestingly, in E. coli, it's just the opposite. Glucose suppresses it. Growth conditions do affect things a lot. I would say our best condition for production is really on solid media. That seems to promote it the most.
And on your list of organisms that make it, you had a bunch of pathogens and non-pathogens. Do you think that the degree of acetylation could be different between whether or not they have to deal with the immune system?
Well, so far, every time we've isolated PNAG, it's been very highly acetylated. The lowest we've ever seen is about 60%, but most of the molecules are greater than 90% acetylated. Again, there probably are environmental or other growth conditions that affect that, but we don't really see anything very low, and we've looked pretty hard for that. It seems to be mostly highly acetylated when we can check that by chemical analysis.
Right here.
Thanks. When you made your vaccine for production, did you test varying the polysaccharide chain length?
Yeah, so we published a paper a few years ago where we looked at polysaccharides of 5, 7, 9, and 11. We compared 5 with 9. We would have done 11, but it was just much harder to make, so we didn't have that much of it. And the pentamer and the nonamer were pretty comparable in terms of their ability to induce antibodies, comparable titers. So we've gone with the pentamer simply because it's just easier to make, and so far it's looking like it's working okay.
Any other questions? Well, not. I'd like
All right.
to thank Dr. Carroll one more time.
Okay, thank you.